All right, in this next screencast, we're going to finish up our discussion on mitosis. So mitosis part 2. So we talked a little bit about the stages of mitosis. We talked about the mitotic spindle. And now we're going to finish up and complete mitosis and cell division by talking about cytokinesis. Okay, so mitosis is division of the nucleus, and cytokinesis is division of the cytoplasm. So once mitosis has reached telophase, cytokinesis is initiated. So, and cytokinesis occurs by a process known as cleavage. Okay, and a cleavage furrow is formed in an animal cell, which is a pinching in of the cell membrane, the plasma membrane. But in plant cells, because of the cell wall, they can't pinch in, so a different structure called a cell plate forms. We're going to look at both of those. So in an animal cell, this would be the cleavage furrow right here. And what happens is a structure called a contractile ring forms around the equator of the cell between the two nuclei, one nuclei, two nuclei, and begins to contract. And if you think about, like, everybody has those cinch sacks, right? And if you pull the strings on your cinch sack, the opening contracts, and that's exactly how the contractile ring works. And it just cinches down until basically you squeeze the center of the cell into two separate pieces, two daughter cells. In plants, cytokinesis is a little bit different because it can't squeeze because of the cell wall. So what happens is at the equator of the cell, between the daughter nuclei, vesicles will gather and they contain materials that will form something called the cell plate. And what they are is basically chunks of cellulose that are going to form a new cell wall. And the vesicles fuse with each other, and you'll get something called a cell plate. So it's a piece of new cell wall contained within a vesicle. The vesicle becomes part of the membrane of the cell, and eventually the cell plate forms in between the two daughter nuclei. Okay, here's a look at, we looked at the stages of mitosis in animal cells, and here is a micrograph of the stages of mitosis in plant cells. So here we have prophase. You can see the chromosomes beginning to condense. Prometaphase, we have the mitotic spindle attached that you can't see. You can sort of see the spindle a little bit right here and right here. But the mitotic spindle attaches and begins to move the chromosomes around. Once the chromosomes are aligned at the center of the cell, at the metaphase plate, you'll have metaphase, anaphase, separation of sister chromatids. And then telophase, we have our two new nuclei forming. You can see the chromosomes beginning to decondense. And here's the cell plate forming between the two nuclei. Okay. So that kind of sums up mitosis in eukaryotic cells. We're going to look at how prokaryotic cells divide. And because there's no nucleus, and mitosis is division of the nucleus, bacteria and other prokaryotes divide by a process called binary fission. Okay, and in binary fission, the chromosome replicates. Remember, there's only one chromosome in prokaryotes, and it's circular. And then the two chromosomes, the two identical copies, just begin to move apart. The plasma membrane pinches inward, sort of cytokinesis, and that divides the cell in two. So it's pretty simple. So let's have a look. So here we have our chromosome, and it begins to replicate at a site we call the origin of replication. We'll talk more about that when we talk about DNA replication, chapter 17 or 18. Okay, so then once you have your two copies, so chromosome replication begins, okay, replication continues on, and once replication is finished, You have 2 chromosomes. They begin to just move to other sides of the cell. Meanwhile, the cell is elongating. Okay, and once you have the chromosomes on either ends of the cell cell begins to pinch inward and basically results in 2 daughter cells. And that's it, binary fission. It's pretty simple. A little bit on the evolution of mitosis. Since prokaryotes evolved before eukaryotes, it is the most likely explanation that mitosis evolved from binary fission. And if you look at protists, some unicellular eukaryotes, they exhibit types of cell division that are kind of in between binary fission and mitosis. So if we look at a comparison here, so here's binary fission on the top, replicate the chromosome, moves to other ends of the cells, cell pinches off, cell divides. Okay. Dinoflagellates, which are kind of a primitive protist, they are eukaryotes, so they do have a nuclear envelope, but the nuclear envelope remains intact during mitosis. And the microtubules, you see they kind of pierce the nuclear envelope to move the chromosomes around. If you look at diatoms and some yeasts, again, the nuclear envelope remains intact, but now we have the mitotic spindle is contained within the nuclear envelope, and it's starting to look a lot more like the mitosis we all know and love. And then if we get to our eukaryotes, we look at basically our familiar form of mitosis. The nuclear envelope dissolves, and the mitotic spindle moves the chromosomes to either end of the cell. Okay, and that's it for mitosis. The last screencast in chapter 12 is going to look at more detail at the cell cycle and how we control it.